Okay, on to the next part. This is excitation, contraction, couplings. We talked a little bit about this, kind of how you get the electrical signal. And after the electrical signal and the excitation, you get contraction of the cell, or of the muscle. So, previously we talked about how we got it with the neuromuscular junction. Let me just go ahead and pull it back up here real quick. This one. We talked about how the neuromuscular junction, we started getting this signal transferring and moving along our neuromuscular junction, right? It's starting to go outside and spread across the sarcolemma, this membrane, right? And it's actually going to travel until it gets to these T tubules. And you remember on either side of the T tubule, what are those called? Those are our terminal cisternae. The name escaped me for a sec. And our terminal cisternae are part of our sarcoplasmic reticulum, which stores calcium in it. And all three of these two terminal cisternae and the T tubule together form what? A triad. So we get the action potential from that nerve signal. The action potential forms here and it continues to pass along. And it's going to travel down into the T tubule. And along these sarcoplasmic reticulum, along these terminal cisternae, we have these special and these specific receptors, these specific channels and receptors here. And I'm going to jump over to another PowerPoint to show this. So here we had the signal was traveling across the sarcolemma, and then it starts to go down into the T tubule. And we have these receptor and this channel. We have a dihydropyridine receptor, which is a voltage sensor. It's sensing the voltage along the T tubule. And then we also have what's called a ryanodine calcium release channel. Okay, so we have a dihydropyridine. This is also called a DHP. So you might see it written as that. So DHP receptor. And we have the ryanodine calcium release channel. What's the key thing to remember here is that the DHP receptor senses the voltage. So it's sensing the change of voltage when an action potential travels down the T tubule. The ryanodine is a mechanical channel, meaning that when the DHP senses a change in voltage, the ryanodine channel is literally going to move. That's why you have kind of like this spring-like contraption. It's literally going to move and open, allowing for the release of calcium. This is a cool little, let me see if I can get this to switch screens. There we go. Okay, so we have this thing called this calcium pump. This is our DHP, and this is our ryanodine channel. So we have our action potential coming down, coming from along the sarcolemma over here, and coming down. And now the DHP senses it, and the ryanodine channel opens, and calcium pumps out. It continues down. This channel senses it, ryanodine opens, and calcium pumps out. And we have this calcium pump that's going to pump the calcium back from out here. This represents up here. This represents the sarcomere, which we'll look at that next step right after this. But the calcium is released, muscle contraction occurs, and then a calcium pump is going to bring the calcium and pump it back into here. And another protein called calsequestrin is going to hold on to the calcium that's getting stored back into that sarcoplasmic reticulum, or that SR. Okay, so that action potential, the action potential comes down. The DHP receptor recognizes it, changes the voltage, the ryanodine channel opens, calcium comes out, contraction occurs, the calcium pump brings it back in, and calsequestion stores it. Okay? Let me move that back. Okay. So hopefully that makes a little bit more sense, kind of what the purpose and the function of that is. Another thing to note, because we have so much calcium already inside of our terminal cisternae, inside of our sarcoplasmic reticulum, that calcium pump, this guy right here, is actually going to be an active pump. If we think back to membrane physiology, what does that mean? It means it requires ATP because it's going to be pumping calcium against its concentration gradient. You can kind of see, this is another picture just showing this, where the sarcolemma, we have everything, the action potential going across all of this, it goes down into the T-tubule. We have that interaction here, the calcium, and it flows and comes back in, and that's where we need that ATP. So the calcium, when the calcium is released by the DHP and the ryanodine channels, and it looks like here he calls DHP channels and ryanodine receptors. They're the same thing, either or. Just one is DHP, the other is ryanodine. Don't worry too much about like the individual naming and semantics, that sort of stuff. As long as you understand what is actually occurring. Okay, so when that calcium has been released outside of the sarcoplasmic reticulum, it's going to go and it's going to bind onto this protein called troponin. Now, if you remember, we talked about troponin and how troponin actually has 3 different parts to it, right? It has an I part, it has a T part, and it has a C part. The I part is bound to the actin, also known as the thin filament. The T part is bound to troponin, the tropomyosin. And that is this protein right here. And that tropomyosin, remember, is covering up the active binding sites that are on the actin. And finally, C troponin, which is open for calcium to bind. So the calcium that's released from the sarcoplasmic reticulum comes and it binds onto, I don't know why that just deleted, that's weird. Comes on and binds to our C troponin. I guess it. Interesting. It made a note. Okay, interesting. So yeah, so that troponin, the tropomyosin, calcium binds to the troponin, and it's going to cause the tropomyosin to actually change shapes. Or I should say, as the troponin is bound to calcium, the troponin changes shapes, which then pulls the tropomyosin, the troponin-tropomyosin complex. It's going to pull the tropomyosin off of the actin, revealing the actin binding sites. So that troponin-tropomyosin complex changes shapes. The active sites are exposed on the actin, and it is available for the myosin binding. So, and this is another little, little detail type stuff. The names of the different binding sites. So, for example, the myosin binding site is located where? That is located on the actin. And then the actin binding site is located on the myosin. Because it's not saying where it is, but what is binding to it. Okay, so these right here are our myosin binding sites. Once that has occurred, we're going to get into next. Oh, wrong one. We'll get into the actual physical contraction. So once that tropomyosin is off and that binding site is available, and I talked about this a little bit briefly. We talked about the sliding filament theory, and we'll talk about this in more detail in the next video. So at this point, we have our active binding site. We have the signal. The signal has gone into the muscle cell through the T-tubule. Calcium has been released from the terminal cisternae of the sarcoplasmic reticulum. Our calcium has bound to the troponin, C, and that has made a change where the troponin-tropomyosin complex, the tropomyosin has come off and revealed the myosin binding sites, or those active sites where the myosin can bind. And that's it for this one, and we'll see you on the next one.